Good afternoon, students. So in my last class I have explained to you all about the structure of an antibody. So what is the shape of an antibody? It is a Y-shaped structure. Okay? And it consists of 4 polypeptide chains. Okay? Now out of the 4 polypeptide chains, 2 polypeptide chains they are present outside and 2 polypeptide chains they are present inside. Okay, now the 2 polypeptide chains that are present outside, they are shorter polypeptide chains, and the 2 polypeptide chains that are present inside, they are longer polypeptide chains. Okay, now the short chains present outside, they have less number of amino acids, whereas the long chains present inside, they have more number of amino acids. Short chains, they have less number of amino acids, so for this reason the chains are light. The long chains, they have more number of amino acids, so for this reason the chains are heavy. Okay, and as because there are 2 long chains which are heavy chains and 2 short chains which are light chains present, so for this reason an antibody is represented by what? H2L2. So all these I have explained in my last class. Moreover, I have also explained that all the 4 polypeptide chains, they are connected together by what? Disulfide bonds or disulfide linkages. Okay, so there are disulfide bonds between the short chains and the long chains, and also there are disulfide bonds between the 2 long chains. Okay, and the long chains, they have a flexible region, and that flexible region is called the hinge. Okay, so it is called the hinge. So upper part is called, the upper part of the long chain is called the arm, and the lower part of the long chain is called the stem. So arm and the chain, they are connected together at a point which is called hinge. Okay, so this hinge, it constitutes the flexible part of the antibody or of an antibody molecule. Okay, now moreover there are also disulfide bonds present within the same chain. So these, you see, they are what? They are the disulfide bonds that are present within the same chain, and all these disulfide bonds present, they provide stability to the antibody molecule. Okay, now I have also explained that each of the chain or each of the polypeptide chain consists of 2 parts, the upper part and the lower part. Okay, this upper part is the anterior part, lower part is the posterior part. This upper part, it is called variable region, whereas the lower part is called constant region. So variable region, constant region. The one that I have given the green shading, that is the variable region, and the one that I have given the red shading constitutes the constant region. The variable region, you'll find that the amino acids sequence will be different, whereas in the constant region, the sequence of amino acids will be constant. Okay, so for all the antibodies, you will find that the variable region contains the antibodies in which the sequence contains the amino acids in which the sequence will be different, whereas the constant regions amino acids, the sequence will be same. Okay, the variable region, it constitutes antigen binding fragment, whereas the constant region constitutes the crystalline fragment. Okay, now why the variable region is called antigen binding fragment? Because this region helps the antibody to get attached with the antigen. So these two shaded structures that I have drawn, they constitute the antigen. And you will find that the antigens, they get attached to the variable region of the antibody. Okay. Now the constant region, it constitutes crystalline fragment because these parts of the antibody, they can become crystalline, they can form crystals. So for this reason, the constant region, it is called crystalline fragment. Okay. Now in the variable region, you will find what there is an area present, and this area, it constitutes antigen binding site. So this will be the antigen binding site. Okay, and to this antigen binding site, the antigen will get attached. So once the antigen gets attached, the linkage will be just like lock and key. So just like lock and key, the antigen will also get attached with the antibody. So just like a key getting fitted to a particular lock, a particular key getting fitted to a particular lock, similarly the antibody will also get fitted to a particular antigen, just like lock and key connection. Okay, now this antigen binding site, it is also called what? Paratope. Okay, so I have once again explained about the antibody because this is very important, and you practice this diagram of antibody very nicely because it usually comes in the exam. So very nicely you practice the structure of an antibody with the labeling. So whatever labelings that I have given, you study it very nicely. Okay, now let us study about the types of antibodies. So types of antibodies. So there you see here how many types of antibodies are there. So before this, first of all, you have to know that what are antibodies also known as? Antibodies are also known as the immunoglobulins. Ig is the short form of immunoglobulins, so Ig. Okay, and there are how many types of antibodies present? There are 5 types of antibodies present. And what are they? They are immunoglobulin G, A, M, E, D. Okay, immunoglobulin G is what? It is called gamma immunoglobulin. Immunoglobulin A is called what? Alpha immunoglobulin. Immunoglobulin M is called mu immunoglobulin. Immunoglobulin E is called epsilon immunoglobulin, and immunoglobulin D is called delta immunoglobulin. Okay, now the first one, immunoglobulin G, this constitutes about 80% of the total antibodies that are present in our body. Okay, so you may get a question that which immunoglobulin or which antibody it is most abundant antibody, or name the most abundant antibody, you are going to write immunoglobulin G. That means gamma immunoglobulin. Okay, so and moreover, you have to remember another point which I have already explained in my last class. That this kind of immunoglobulin, it can pass through the placenta from the mother to the newborn baby or to the fetus. So from the mother, this immunoglobulin can pass through the placenta. It can pass through the placenta into the newborn baby or into the fetus. F I am writing, it is fetus. F-O-E-T-U-S. Fetus. Okay. So, this gamma immunoglobulin constitutes 80% of the total antibodies present in our body. Okay. And this kind of immunoglobulin, it is very important, and it passes from the mother to the fetus or to the newborn baby through the placenta. This you remember, you may get a question on this. And it is present where? It is present in the blood, it is present in the lymph, and it is present in the intestine. So it is present where? In the blood. It is present in the blood, it is present in the lymph, as well as it is present in the intestine. Okay. Now the next comes immunoglobulin A, which is called alpha immunoglobulin, and this constitutes 10 to 15% of the total antibodies that are present in our body. Okay. The most abundant is the immunoglobulin G, and the second most abundant will be immunoglobulin A, that is alpha immunoglobulin, and it constitutes 10 to 15% of the total antibodies present in our body. Okay. Now, where is this immunoglobulin or where is this antibody present in our body? So, this antibody is present in the sweat. It is present in the tears, it is present in saliva, and it is also present in colostrum. So remember, colostrum, colostrum. It is present in the colostrum of the mother's milk. Okay. So I am repeating once again, where is this immunoglobulin A present? It is present in the sweat, tears, saliva, mucus, colostrum, and also even gastrointestinal secretion. Okay, so from here you may get another question, that which immunoglobulin or which antibody is present in the mother's milk? It is the immunoglobulin A or antibody A is present in the mother's milk. And in which part of the mother's milk it is present? It is present in a yellow color substance of the milk, which is called colostrum. So already about this I have explained in my last class. Okay, so already I have explained about this in my last class. Now the next one is now, it is also present in small amount in the blood as well as the lymph. So it is also present small amount where? In the blood and lymph. So this is also present, the immunoglobulin G, this is also present in blood and lymph. Moreover, this immunoglobulin A is also present little bit in the blood as well as lymph. Okay. Now the next comes the immunoglobulin M, and this immunoglobulin is called mu immunoglobulin. Mu immunoglobulin, and this mu immunoglobulin, it constitutes about 5 to 10%. So about 5 to 10% of this immunoglobulin is present in our body. Only about 5 to 10% is present in our body. Okay, and the largest immunoglobulin, that it is the largest immunoglobulin. Remember, it is the largest immunoglobulin that is present in our body, and it is found in lymph. It is found in lymph. Okay, and remember, the earliest immunoglobulin to be synthesized by the fetus. So it is the earliest immunoglobulin to be synthesized by the fetus, and moreover, it is the largest immunoglobulin that is present in our body. Okay, so I hope you have understood about the immunoglobulin M. Okay. Now the next is the immunoglobulin E. So immunoglobulin E, this is called what? It is called the epsilon immunoglobulin. It is called epsilon immunoglobulin, and it is present in a body about 0.1%. So less than 0.1% of this immunoglobulin is present in our body. Okay, but this kind of immunoglobulin, it is very important immunoglobulin. Why? Because it is present where? It is present in the mast cells and basophil cells. Okay, so it is present in the mast cells as well as the basophil cells. Okay, and moreover, this kind of immunoglobulin, it is produced in large quantity in case of allergic person. So whenever you suffer from allergy, you will find that your immunoglobulin E, it increases. Okay, so this immunoglobulin E, though it is present very less quantity, it is less than 0.1% is present, but this kind of immunoglobulin, this will increase in our body during allergic conditions. Okay, now the last one is immunoglobulin D, which is also called delta immunoglobulin, and this delta immunoglobulin, it constitutes 0.2%. So 0.2% of delta immunoglobulin is present in our body. Okay, so these are the different kinds of immunoglobulins that are present in our body, and you study this very nicely. You remember the names, what are the different kinds of immunoglobulins that are present in a body, and moreover, you may get questions like immunoglobulin G. This kind of immunoglobulin, it is the only immunoglobulin that can pass from the mother to the newborn baby or fetus through placenta. Another one, remember immunoglobulin A. This is also very important because it is present in the mother's milk. It is present in the colostrum of the mother's milk. Okay, you also remember the immunoglobulin E, which is called epsilon immunoglobulin, because this immunoglobulin, though it is present in less amount, but it increases during the allergic conditions. Okay, so I hope you have understood about the types of immunoglobulins. Now the next comes the immune response. So immune response. Now first of all, you have to remember, what do you mean by immune response? Okay, now when an antigen enters inside your body, antigens means what? Germs. So when any kind of germ enter inside your body, the germs will act just like a foreign substance. So your body will try to get rid of the germs. Okay, your body will try to get rid of the germs. Okay, so the body will react against that germ. So that reaction of the body against the germ, it is called immune response. Is it clear? I am repeating once again. What do you mean by immune response? when an antigen, that is germ, enter inside your body. Antigens are what? These are foreign particles. These are foreign substances. These substances, when they enter inside your body will not adjust with the foreign body. So what your body will do, your body will try to get rid of that foreign substance. So how your body will react to the foreign substance? Your body will react by producing some kind of substance, so that those substance will destroy the germ that has entered inside your body. So this reaction of your body to get rid of the germ is called immune response. Okay, now immune response, you see, it is of two different types. The first one is primary immune response and the second one is secondary immune response. Okay, so what do you mean by primary immune response? So when your body comes in contact with the germ for the first time, so when for the first time your body comes in contact with the germ, the reaction that will take place in your body will be primary immune response. Is it clear? But when your body will come in contact with the same germ for the second time or subsequent times, okay, the response that will be produced by your body will be secondary immune response. So I'm repeating once again, what do you mean by primary immune response? Primary immune response means when your body will come in contact with the germ for the first time, the reaction that will be produced by your body, that will be primary immune response. And when your body will come with the same antigen, with the same germ for the second time or subsequent time, the reaction that will take place in your body will be secondary immune response. Is it clear or not? So the primary immune response, you'll find the action will be very slow. The action takes a longer time. Why? Because your body will, when the germ will enter inside your body slowly will produce the antibodies. So immediately your body will not produce the antibodies. So antibody formation in your body will be slow. So action of the primary immune response, it is a slow process. So action takes longer time. Okay, but in case of the secondary immune response, you'll find that the response will be very fast. Why? Because memory cells